Northern Tracy's Scribblings. The Amino Age and the New Abnormal Doctors. mRNA is apparently the only new cure for all our ills. They're even giving this new era in medicine a name, calling it the Amino Age, but is it all it's cracked up to be? I just heard too that anti-vaxxers are trendy now, which tells me they are ready to complete their big swap of vaccines for gene therapy. Problem, reaction, solution in full swing. The Amino Age is the offered solution, but is it a solution at all, or just another terrible healthcare paradigm to keep the slaves manageable and controlled? Are you confused by all this talk of messenger RNA, proteins, genes, genomes, DNA, and contagious disease? Well, you're not alone, and you are being confused on purpose. The way out of the confusion, I thought, was to dive in, swim around, and see what I can find that is solid and factual in all this science waffle being foisted on us. People with bits of paper to say they are qualified to tell us the facts are either lying or also confused. I spent 3 weeks looking into their papers and articles, methods and tests, and gave myself a massive headache. It reminded me of the days I was trying to argue with shills over vaccines and what they did and did not do, which actually taught me a lot about their so-called science and how they confused people with their fancy language there too. All the technical stuff I learned about back then is just being rehashed and plunked onto what we are told about RNA and DNA. Just swap the word virus for DNA RNA and you will see the same exaggerations, assumptions, and lies. I have to hit you with some big misconceptions which pull the rug from under the feet of modern medicine. This is a bit scary for some people, but I promise you the truth is far prettier than the convoluted, ugly theories we've been sold. First bombshell incoming. Does it exist like the pretty pictures say it does? The theory of a blueprint in the nucleus of every cell. As I was trying to knit all this info into some sort of semblance of cohesion, I suddenly wondered, where were the chromosomes in all this? No one mentions them or what they are, so I looked them up. Supposedly DNA is wrapped up in the chromosomes in the nucleus of our cells, which have only ever been seen when cells are dividing, only seen in embryotic cells, in other words. They see these chromosomes double and split into the new cells as it divides into two. This process is called mitosis. DNA cannot be seen though, and the very few photos of what we are told is DNA are very dodgy. 2 I looked for have even been deleted from the internet, but I found them on the Wayback Machine. Why did they delete them? Probably because they look so fake. Also, sadly for them, they look nothing like their cartoon DNA. What did Lanka have to say about DNA and RNA back in the 90s?
I already had a somewhat critical attitude when I started studying molecular genetics, so I went to the library to look up the literature on HIV. To my big surprise, I found that when they are speaking about HIV, they are not speaking about a virus. They are speaking about cellular characteristics and activities of cells under very special conditions. I was wondering what viruses are for in evolution, because they didn't seem to have any function other than to be very dangerous and killing other cells. So I went into evolutionary biology and found that the first genetic molecule of life was RNA, and only later in evolution did DNA come into existence. Every one of our genomes, and that of higher plants and animals, is the product of so-called reverse transcription, RNA transcribed into DNA. But I had already realized by then that the thinking about molecular genetics was very dogmatic. In the early 1960s they came up with the central dogma of molecular genetics, which they try to uphold even today, and which is ridiculous. The dogma says that DNA behaves in a static way, it doesn't change. DNA makes RNA. RNA cannot be transcribed back into DNA. RNA comes into existence only on the basis of DNA. That was and is the basis of the central dogma of molecular genetics. While studying the evolutionary aspects of biology, I quickly realized that reverse transcription is common to all forms of life, and in fact is the basis of all higher living. Later I learned that reverse transcription is a repair mechanism for chromosomal DNA. But the mainstream of molecular genetics is still committed to the central dogma. There is no such thing as reverse transcription from RNA to DNA. In 1970, when they detected biochemically that there is a reverse flow of genetic material, they didn't give up the dogma or even try to change it. Instead, they called it an exception to the central dogma of molecular genetics, and explained it by postulating the existence of retroviruses.
From Mark Gabrish-Conlin interviews Stefan Lanka from the December 1998 issue of Zanger's magazine.
We are told that our chromosomes, the genome, were mapped by a man called Morgan, but all I can find is work on plants and insects. How did he map the human chromosomes by looking at plants and insects? They used classical genetics, as in looking at natural mutations like Mendel, a botanist, did with his peas, which all this stuff is based on. Peas with different colored flowers. Inbreeding and crossbreeding. Chromosomes are like your sock drawer for keeping all those 6 feet of strings of DNA in some kind of order. But it's not just one string of DNA, because we have 23 paired chromosomes making 46 in all. So that's a lot of blueprints, not just one. Oh, and guess what? We can see chromosomes under a normal microscope, but what they are and do is something else, because they are only ever seen during cells dividing, as in fetal cells. If that's the case, there's no proof they are in all cells, and Harold Hillman disputed this. More recently, new pics are emerging saying they don't look anything like the pictures we've been sold again. The strands of DNA are supposed to be wrapped up in a globular mess somehow. Looks like they've been looking at bacterial chromosomes, or is it just more debris, and now they're fighting over which ones are the real deal. The new cartoon drawing of a chromosome looks just like a picture of their proteins. Like a lump of badly tangled wool. I can't help but say too, even though I might get blasted for it, that the original chromosomes look an awful lot like bacteria. There, I said it. Then I remembered this. From an old article I edited by a fellow independent researcher called "Is It Time to Give Up on the Flawed Modern-Day Science of Genetics?" by Lee Stevenson. They will not consider the fact that humans, animals, and plants have microbes that modulate their genes. Changes in the functions of those genes also change animal and plant microbial makeup. Microbiota can alter host gene expression. These things have definitely not been considered by the biotech industry or the pharmaceutical industry. Something as simple as altering our diet can change our microbiome and alter gene expression in the body. So, are the genes small strings of protein making recipes really coming from our own cells, or are they from our bacteria? Do our cells make the proteins from reading instructions from our own blueprint DNA, or do the bacteria deliver them? Seeing as there is no proof whatsoever of our own cells doing this, and the fact they are using bacteria and yeast to produce synthetic proteins, it looks like it's all a lie, a mistake, or assumptions. All the work I looked at about producing synthetic RNA DNA seems to use bacteria and yeast to produce the product, which, by the way, a company called TriLink, who produce it, call it RNA, a drug. Funny description for something which is supposed to control our whole existence and heredity. On the production end, I just saw mixing of chemicals, acids, and catalysts with yeast and bacteria, and then the usual poisoning and centrifuging plus heating and cooling till the end product pellets, called synthetic RNA, is left, and they call it a drug. This is what they call biochemistry. Is it producing chemicals from biological processes, or is it condensing living biology down to chemical processes, or a bit of both? Just as they did with herbal medicines, plants, to turn them into patent-able drugs. Just for clarification, we need to look up some of the things they talk about in these gene modification processes. RNA is said to be just strings of acids. In their CGI illustrations, they look like half a string of DNA. How do they know it has a function, and how can it pass messages if it is just a string of chemicals, acids? What are acids? Acids. So an acid is a reactive substance, not necessarily negative. Sounds like what an enzyme does too. So in this context, acids don't mean acids as an acid alkaline, okay? So what is an amino acid? Any chemical substance found in living tissues then. But then look what it says about protein too. So proteins are just a mixture of chemicals too? Well, in their eyes they are. So the supposed spike protein is a bunch of chemicals mixed in a certain way according to them. Sounds just like a drug, doesn't it? Now maybe we can understand how they might seem to make them in a chemistry lab. There are so many scare stories coming out left, right, and center about how much damage the spike protein could cause, and not only to those who take the shots but maybe even those close to them who haven't, so raking up the old contagion myth once again. We know viruses are a lie based on germ theory, another lie, but now we are made to believe proteins can suddenly spread disease person to person by shedding of a fictional protein, which they say they can force our own cells to produce at their bidding. But can they? They are mixing chemistry and mathematics and applying them to biological functions. This is the materialistic world these scientists live in. We are just breathing, walking chemical labs. We need to look at how the science of genetics has evolved, seeing as it is becoming the new dominant science in medicine. They brag about it replacing chemo and vaccines. Well, could it be any worse than chemo? Could it be any worse than vaccines? Or is it just the same stuff repackaged with more bells and lights? The processes in the lab look like smoke and mirrors to me, all to hide the fact they either do not know what they are doing apart from chemical mixing, or they do know what they are doing but don't want us to know. In fact, it looks like the alchemy of old, which was also very secretive, and the old science of the Freemasons. Let's have a look at the evolution of the smoke and mirrors. Classical genetics was a very slow process of damaging, poisoning, and observing reactions, mutations, and earlier just inbreeding. Classical genetics looked for new mutations in plants, peas originally, and fruit flies because they reproduce very quickly. When they saw a mutation, they blamed the genes. Genetics was all about inheriting traits, but it has morphed into genes causing changes in real time. Just like they blamed germs and then viruses as scapegoats for chemical poisoning or malnutrition, they have gone now to molecular levels of invisibility. So a plant grows with unusual features, and instead of asking what caused it to grow like that, they claimed the reaction as the cause again. As with the TMV, they assumed it was something smaller than a germ, so a virus, when it was soil depletion, malnutrition. Just as pleomorphism happens and bacteria morph to survive a hostile terrain, the morphism is not the cause, it is a symptom. The terrain is bad. Everything has to adapt to its environment or perish. In the 90s, they went from classical genetics to technology and invented the microarrays. Basically, they take the cell debris and put each bit robotically onto a chip arranged like thousands of jigsaw pieces, just like you would spread them out to see before you start doing the jigsaw. Except they claim to know what each bit of cell debris is and where it is placed. Like this. All very nice, but the reactions they claim are from specific genes were identified by classical genetics using observations of reactions to terrain anomalies and lots of guesswork. Now the assumptions made have become colored dots produced by computers, robots, and chemical reactions, all programmed into computers from their old poisonings, observations, and guesswork. These assays were adopted from gene labs into virology labs since they announced that viruses were bits of genetic material, so we can see how the two sciences were already crossing over. All the CGI pictures of DNA and talk of snipping and editing makes it sound very high-tech and mysterious, but do you know what they use to snip and join these supposed strings of acids? Bacteria. Yeah. Apparently E. coli is a fave tool for snipping at amino acid strings they call RNA. The same bacteria that dismantle proteins in dead tissues in our guts and also claim are very pathogenic and cause sickness. More proof that bacteria are indeed the janitors in the body and they know it. In fact, it exposes they might be even more than just janitors, doesn't it? What else do they say about RNA? According to dictionary definitions, it is a polymer. It is some monomers clumped together. Monomers are another chemical compound which may or may not react with others. Plastic is a polymer, so is paper. All the info you find about these polymers and reactions between proteins and acids when you look them up separately are chemical and atomic reactions. Like this. It is a picture of a chemical reaction, yet it also looks eerily like what they claim goes on at molecular level in the body. This is chemistry overlaid onto biology. That polymer looks exactly like their description of a virus, doesn't it? Coincidence or plagiarism? A bit like saying all birds are aeroplanes because we can understand how aeroplanes work. We are supposed to believe the inside of our cells looks like a plastic factory, in other words. So let's have a little look at how they claim to do all this gene editing and splicing, which looks so high-tech and clever in their cartoon pictures. They are claiming this Cas9 is a protein from a bacteria's immune system. Now that is interesting, isn't it? They claim that bacteria produce proteins based on past experience of damaging chemicals, just like the story of our own immune systems do. So who is driving this immune system? Is it our cells or our bacteria? Could it be that bacteria are not only our janitors but they are also our immune system, instead of being the enemy that our immune system was said to attack? This little nugget just tells us that they knew all along bacteria were helpers and not enemies. They clean up and they recycle cell fragments, and they are trying to harness that. Problem is they only do this in our body with dead cells and debris. They do not harm or damage healthy living cells. Remember, RNA-DNA is not a living substance, just like viruses were not. Are they trying to make this protein work for them on live tissue cells using gain of function? There's been a big hoo-ha about gain-of-function experiments, and we are led to believe they have been trying to make a virus. But what about this? No mention of viruses here. Is the gain-of-function story a bending of the truth hidden in plain sight? Are they trying to make these proteins edit genes in live cells and live subjects, us? Trying to genetically modify us under the guise of curing something? Remember what Lanka said about reverse transcription. There are lots of indications to tell us they have been failing dismally in their efforts, though. They've also been hit with public outcry. Did you know that back in 1974 all genetic engineering experiments were banned because of public concern as to where it was leading? The genetic industry held a conference and declared to the world that they were just trying to find cures for deadly diseases like cancer, and this was the only way. They were only allowed to resume work on the back of this massive promise, which was a lie. Now we have gone from banning their experiments on fetuses to all volunteering to be their human guinea pigs, and the world is their oyster. I found this story after I published but want to add it in now as it shows just how bad this wall was that GM was said to have hit. Jesse Gelsinger was 18 years old when he volunteered for a clinical trial at Penn State to test the effect on GT on a rare metabolic disorder called OTC deficiency. Within hours of being infused with corrective genes encased in weakened adenovirus, Jesse suffered multiple organ failure, and days later, his blood almost totally coagulated. Swollen beyond recognition, and brain dead, he was taken off life support. His death caused the then booming field of gene therapy to grind to a quiet screeching halt. When I went to Penn, as my first stop on the interview tour for the talk article, the head of PR there said, "Not sure what to tell you. We killed an 18-year-old kid. This article is well worth a look." From the complete guide to understanding CRISPR gene editing. So only the last 5 years have human trials been allowed, and 2020 gives them their results. Is COVID their big human genetic engineering trial? Obviously yes. Who gave them this permission to break all the rules made back in 1974, to allow them to continue? Is this why they needed an emergency to bend those rules? I think so, and that is why COVID was rolled out in the first place, but if you think this is the first time they've used this technology, you'd be wrong. The first vaccine they produced in a gene lab was the Hep B, hailed as the first cancer vaccine. The very one that caused the disease they called AIDS, which Fauci and Mikovits were in on. The next one was the HPV, which was their first attempt at curing cancer, except it didn't cure anything, did it? They can't cure cancer because they know it is not a disease, so they plumped for the unprovable prevention route, and this was their second attempt at a cancer vaccine, with dire consequences again. Let's regroup again back to the cell debris. We are told the mRNA is a short strand of genetic material which tells the cell how to mix all the amino acids to make a specific protein. If we have a blueprint, why do we need these smaller bits of info, and where do they supposedly come from? If they are just a strip of different chemicals, how on earth do they do all the things they claim? From what they are claiming, it's like saying a snip of a hair from my head can do double and triple salcos while cooking the dinner. They are discovering loads of more supposedly different RNAs too every day. Just add a letter or two before RNA, like an M for instance. The inside of a cell is starting to look like downtown Manhattan by the sounds of it, or are they simply looking at loads of cell debris from cells killed by their processes in the lab? And playing a weird guessing game at who can come up with the best story, explaining what it might do. I found this gem of a paragraph in a Science article.
For many new ncRNAs that are still being discovered, the function seems a bit more questionable. Many scientists are questioning whether assuming function because of existence is a Panglossian. Some of these RNAs might just exist as a byproduct of another process.
Guess what Panglossian means? Extreme optimism. Yes, it does look like science is fooling itself. Apart from everything they've seen of cells under the totally alien landscape of an electron microscope, these cells are always grown in tissue cultures. Harold Hillman pointed out that the cells in a tissue culture have significantly different morphology, biochemistry, and environment than the cells from which they came. Meaning, all in all, they might as well be looking at a lump of dog turd to describe the workings of a human cell. None of it can be believed, because the fantastical story they have concocted goes against everything we see in nature, which is always simple and perfectly sensible. Harold Hillman tried to stop this nonsensical storyline back in the 70s, providing evidence that our model of the cell was completely incorrect, and used an old proverb as the intro to his book.
It is easier to ride on the back of a tiger than to climb off it.
This Russian proverb, which appears at the beginning of Dr. Harold Hillman's last ever book, sums up the current state of our scientific establishment. Whether medicine, physics, or archaeology, all facets of scientific research rest on certain dogmas. In essence, what this proverb is saying is that it's easier to carry on perpetuating falsities than it is to admit you were wrong, drop everything and start again.
In my 3 weeks of delving into genetics, I found no evidence of any of these things RNA is said to do. Since the PCR and assays were invented, these are all that is used, along with chemical reactions, a lot of maths and computer programs, all based on the original findings back in the early 1900s, which looked at mutations and changes in fruit flies. Along with all the high-tech gear and AI, they have moved from dead tissues to bacteria and fungi to do all the work for them. If they don't need the cells to do the gene splicing, then surely it follows that it is the bacteria themselves that do it in the cells too? Bacteria are all killed when looking at cells under an electron microscope, just like everything else, so they're claiming the bricks and mortar puts the fire out in a building and not the fireman. To use the old analogy again, apparently cells with no nucleus, so no DNA or chromosomes, like platelets in the blood, can produce proteins too. Where do they get the recipe from? In the science labs working on genetics, the only biological ingredients they use are bacteria and fungi yeast. Remember, fungi and yeast are just another form of the bacteria, in the pleomorphic stages. They make genetics look very high-tech with talk of snipping and editing, but remember, bacteria recycle cell debris. They also produce enzymes, which are a type of protein. which cause the different amino acids which make up the proteins to break apart, just like they do in the digestive tract. So from all this they deduce that RNA is as inanimate as DNA, because it is just strings of acids, it doesn't do anything, the bacteria are doing all the work in the lab, and in the cells. Other things I saw mentioned a lot in the gene lab work was something called plasmids. They tell us that these are bacterial chromosomes. They use them as vectors to deliver drugs RNA into cells. The more I looked into these plasmids, the more convinced I became that they were talking about bacteriophage again. They base the whole of virology on these phages, remember? So plasmids are produced by bacteria and look like their description of a virus, and they contain some genetic material. Bingo! Plasmids are viruses are bacteriophage, and let's go one step further, because in the pleomorphic graph, what is the start and the end of the bacterial morphology? The somatid, also called the microzyma by Beecham. It is immortal and contains all the information to start the whole process of life. I've hinted at what I think this RNA could be already, but then someone sent me a book in French about Becham's work and I translated this.
Bacteria are remarkable machine tools, intended to dismantle old sick cells, cancers, or scaffolds used to repair certain injuries. In this sense, it is fundamental to understand that we do not get germs from the outside. There is no biological warfare, says Bechamp. In an alkaline environment, microsms assemble and manufacture bacteria. But if, on the contrary, the environment of the body returns to a neutral pH, the bacteria disassembles and the microsms recover, moving freely. Thus, depending on bioelectronic constants, temperature, presence or lack of oxygen and nutritional substances in the living environment, microsms communicate with each other to form a certain germ, a mycelium, a microbacteria that will be capable of fulfilling a certain mission. A body does not get infected by contagion. Bacillus or bacteria are built on the spot, when needed, to destroy damaged cells or tissues and to evacuate waste. Diseases of so-called pathogenic germs are healing processes. They don't become serious unless important nutrients are missing. For example, in the case of tetanus, microsomas build tetanic bacillus in deep muscle injuries caused by wounds or burns in an anaerobic environment, no oxygen. Their mission is to evacuate damaged cells and to rebuild new tissues. But this work requires a lot of energy and generates waste. In the absence of vitamin C, these wastes becomes toxic and trigger the famous spasms of tetanus. It has been shown on several occasions that IV injection of magnesium chloride stops the spasms in half an hour. Alternatively, the injection of vitamin C brings healing in 2 or 3 minutes. Microsomas are absolutely remarkable considering they can transform into other forms of living matter. They are complementary to each other, autonomous, specific, intelligent, and responsible. They always do what's best for the body. But for this we need to provide them with the elements indispensable to their lives and their functioning. Air, food, vitamin C. Microlyses, for example, E. coli are capable of reproducing at very high speed and in very large numbers. They are not invaders or war aggressors. They feed and metabolize certain substances through a digestion mechanism. They need sugar, proteins, fatty materials, and trace elements. They are living entities. They dismantle and remove waste. These entities made by microzyme can be dismantled to atomic chemical level again. Microzymas are almost eternal except for brutal destruction, through extreme processes, cremation, soaking in formaldehyde or pure acid. They can go into hibernation, partially dehydrated. Thus, live microzymes were discovered in fossils 12 million years old. Microcytes are specific to each individual and are likely responsible for the transmission of hereditary characters. Properties of microzyme sickness, a rehearsal for life microzymes. Antoine Bechamp.
Right. So now we've sorted out that whole mess, let's look afresh at the mRNA shots and the COVID story. The big topic on everyone's lips now is the spike protein. We know they have no virus, so there is no spike from any imaginary virus. This one fact that we know there is no spike protein in the shots makes these shots a fraud. They are using the word vaccine as a cover story and claiming the gene shot works like their vaccine theory, because they can then be protected under the Vaccine Act from any litigation for the damage they are causing. Doubled with that, they get to conduct their massive experiment on the back of an emergency pandemic rule, permitting an experimental vaccine for a made-up disease. This is double fraud. They really have pulled out all the stops this time. They are saying the spike protein is a poison toxin. We discussed Arthas last time and his dabbling with poison proteins. So what are they? When Arthas was playing with them, they were venom from snakes, and later Richer played with jellyfish venom. They were playing with injections of these poison proteins. The supposed deadliest of all poison proteins is said to be botulinum toxin. So how come people volunteer to have this stuff injected into their faces regularly? If it's so deadly, why do they not drop dead? It does seem to paralyze muscles locally, but doesn't spread and kill the whole body. Neither do they shed proteins, so causing others to have instant facelifts. Ingested protein poisons only seem to affect the digestive system, so it would seem the poison is localized and does not spread like a so-called virus is supposed to. Even though they claim extreme deadliness, they can only affect the direct tissues they come in contact with. So far so good. Could this be what their gain-of-function experiments have been trying to fix? I delved a bit deeper into poison proteins but couldn't find much more of interest, so ended up on general poisons and looking at this website. I found this paragraph. Some of this is obviously assumed, and cause and effect being swapped around, but basically they're saying that some poisons can cause mutations. Yes, we saw that with thalidomide, but what does it have to do with DNA? Nothing, as these mutations do not get inherited. People damaged by thalidomide can have perfectly formed babies. How do poisons mimic? They obviously don't, so let's say they cause a reaction which damages the cell. Much simpler. Electron loving would imply a change in pH would occur. This would affect proteins produced. I didn't know what electrophilic meant, so clicked on it and Lou key here. Lots of those poisons are pretty common in our day-to-day lives, I'd say, so maybe we can conclude that poison is not only in the dose, but also in the vicinity. Maybe like weeds, or just plants, wild herbs, growing in the wrong place, these chemicals only become poisonous when they are in the wrong place, as well as how much of it gets there. We also know we can become tolerant to an extent to these poisons in small doses. But not to proteins? Why are they treated differently? Remember the anaphylaxis process. We still never worked out why that happens or how. I also found out proteins can change their shape and function according to the environment, and it seems to have a lot to do with pH and electrical charge. The pH measurement is actually all about the positive or negative charge of things. So the optimal pH is slightly alkaline, which is a slight positive charge. Even though the sciences know this, the biology side largely ignores it and treats the body like a chemical soup. It makes me wonder if all that junk DNA they can't understand is nothing to do with recipes for proteins and chemicals and where the real stuff is. I'm reminded of a scene from the film The Fly, where the scientist has to try and program the essence of life into his computer, and his girlfriend tries to explain it to him, but can't either. Back to the lab. So this spike protein that they want our cells to manufacture, was it to be a poison protein? Well, we know for damn sure it didn't come from a virus, so where did they get the idea for it? From poison proteins and Arthas and anaphylaxis? Did they make it? Can they make it? From what Malone told us, yes, they have a synthetic protein on the shelf, so we can only guess what it is hoped to do from their objectives. Sterilization seems to be their main aim, and it just came out from autopsies that this protein is being found concentrated in dead subjects' ovaries, at least that is what they claim. Are they making a protein which is a specific poison to specific cells? That would make sense, so it would end up where it's supposed to be, right? How do they know it is concentrated in those parts of the body? What test are they using to show this? PCR or assays? Both bogus tests. Are they finding a specific protein in these places, or is it, as always, the damage caused there by the poison protein? They have been swapping the words virus and protein for some years now, and their evidence of a virus is always the damage caused by their chemicals. So it would follow that the same evidence is being claimed here too. What is this thing off the shelf? From what I gather, it's a string of letters and numbers from the gene bank, which is more or less just hundreds of recipes for making synthetic proteins. Only our own proteins, made by our own cells, are welcome in the body, it would seem. So even a human protein, from someone else, would be foreign. How does the body deal with foreign proteins? This is where their immune system theory comes into play, and they treat foreign proteins with the same story as germs and viruses. The immune system story. Just like they concocted viruses from thinking they were seeing things exploding out of bacteriophage. I'm beginning to wonder if they are swapping these proteins for what they thought were viruses exploding out of cells, which were actually just dead cells being dismantled. They based viruses on bacteriophages, as I explained in Going Viral, so are they just expanding on that storyline now with proteins? Are proteins to be the new viruses? Messenger RNA is a figment of their imagination too, but let's say that it is a drug, as they themselves describe it, and a mixture of chemicals, which is what all drugs are. This drug obviously can't provide our cells with any recipe to make a protein because it is foreign. It can't get in. Just like in vaccine theory, they need an adjuvant, a poison, to cause any reaction, or rather to blast it in. They need a poison to deliver this poison into the cells. Our cells would never make a synthetic, foreign protein either, as that would go against all the rules of nature. They know this drug can't gain access to our cells, they speak about it openly. This is why they say they used plasmids, or did in the past, to deliver the load. That didn't work either, because their virus theory is wrong, and plasmids are not viruses anyway. They have used the old immunity card too, to pretend they need to cloak the drug so our body's defense system won't see it. The whole story of these shots is geared to pretending they are vaccinating us against a killer virus, and the truth couldn't be further from that. The antibody tests are another smokescreen, long used in the vaccine fantasy. They claim antibodies prove the vaccine works, but antibodies also do not exist. They are globulins and they are nonspecific. That means the old theory that we make specific antibodies to specific germs is nonsense, and they know this for sure because they know there are only a few different ones according to them. They are just proteins which clump together around toxins which can't be processed by enzymes, also proteins. Compare it to throwing a blanket over a fire to put it out. So the appearance of globulins signifies any kind of poisoning which the body is having trouble processing. Not a specific marker for any specific virus or germ, nor any indication of immunity, which was clearly exposed by the HIV fiasco. It seems they've been struggling to put that little nugget back into the box since they let it out. Proof Fauci is a clown. I watched a couple of painful videos, talking to a scientist called Robert Malone, who claims to be the inventor of this mRNA technology. Apart from the obvious chip on his shoulder, over probably not getting a Nobel Prize, his whining does reveal quite a lot about this whole gene vaccine swap game they've pulled. First, I was struck by how little he seemed to understand about biology, as he claimed they thought the ingredients from the shot would stay in the arm. What? He reminded me of Judy Mikovits and her whining about being ousted from the old HIV game and how she exposes more than she thinks. I made copious notes from his interview with Smell Bigtree, but will try to compact so much bullshit into hopefully a few nuggets. He explains that he was working on vaccines at the Salk Institute when a friend in genetics told him they hit a wall. I remember talking about this wall before, and he explains what the wall was. They couldn't get the cells to express the foreign proteins at all. That was the Great Wall genetic science hit. They thought it was our immune system and they couldn't work out how to get round it. They'd tried to fix children with no immune system. Do you remember the boy in a bubble? It's blatantly obvious to some of us that he was vaccine damaged and became a human lab rat for the genetics industry. All they did in the end was give the poor kid leukemia. They also failed miserably with a cure for cystic fibrosis and never could find a way to cure cancer, as they promised, but they sure could induce it. So as I explained in my last podcast, vaccines were struggling particularly at the Salk Institute, as they had killed loads of kids with their polio vaccine and were now persona non grata. And genetics had hit a wall, so these two Muppets thought, why not get together, and we can sneak the non-working gene therapy in through the back door of the vaccine industry. It was a purely financial and political maneuver. Nothing to do with saving mankind. He let slip that protein causes the immunity, so either he is thick or he's gone off script, and revealed that they know there are no viruses, and that proteins can induce an immune response. Remember anaphylaxis. He also went on to state that the shots cause ADE. I already told you, that is science waffle for anaphylaxis. So he exposed they know this too. He said that the spike protein, he talked about spike a lot, like it was a friend of his, is a biologically active protein, it has effects, it modulates the immune system. Now firstly, that last bit is bull, but how can he say this with such conviction, unless he holds a patent on it? Or is it a euphemism for one of the drugs they've developed called Spike? He also stated his friend Spike can open the blood-brain barrier. The best quote for me though was this.
NIH engineered the Spike off the shelf.
Off the shelf implies they got it from the gene bank, where all the patents are, for ingredients to make synthetic proteins, not viruses. And it would have been on the gene bank already before COVID was launched. Here is your made in a lab virus, oops protein. He said these are first-generation mRNA shots, and that no toxicology studies have been done, yet then he slipped in. Mice lie. Monkeys mislead. Only human data is relevant. Which us antivivisectionists already knew, but he is openly stating that COVID is the toxicology study. Wow. Malone is in the business of repurposing drugs out of his own mouth, and works for the FDA, which he said is not independent of political pressure, and Department of Defense. Well, he certainly has repurposed vaccines and gene therapy, hasn't he? I wonder though, how Gates managed to wheedle into this cushy little arrangement. There's no mention of him at all. No, hang on, he did mention Moderna. During the original inventing, he says there were patent battles for this new idea of his. Between Moderna and BioNTech for recipes produced by him at Salk and the others working in the cancer field. There we are, Gates was sticking his fingers in the pie, right up to his elbows, yes. Most of the rest of his babble was just junk DNA, and of course pushing the bogus cure for COVID, being some parasitic poison. So many people are buying that crap too, even if they don't believe in COVID, they think it will placate the believers. All it does though, is send everyone running back to the people who cause all this sickness in the first place, with their poisons. It drives me insane, the pure insanity of it all. A Romanian friend on Facebook wrote this, and it basically says it all in a nutshell.
I don't believe in the bio-nanotech manipulation thing that so-called scientists brag about. What I think is that these vaccines contain certain toxins that have already been tested on prisoners, orphans, mentally sick and homeless people. My opinion is that the top producers and doctors in the so-called research claim anti-COVID vaccines are experimental, just to provide a public excuse for the dramatic side effects occurring in cases of some of the vaccinated. However, the idea that the world's scientists have the scientific and technological ability to manipulate at bio-nano level, in my opinion, is disinformation, so that they can appear in the eyes of the popular masses as semi-gods of intelligence and knowledge. I believe that especially in the biomedical field, all the so-called advances in knowledge of the effects of toxins in vaccines and pharmaceutical medicines are only done through uninterrupted clinical tests on defenseless people. So, how could they reveal this to the public without compromising themselves? So the so-called biomedical researchers misinform and manipulate to give people the impression that they hold astonishingly advanced scientific and technological capabilities, although in reality, with their poisons they cannot heal even a trivial cold. Nikolai Nedelcu.
Compare all this with what Moderna thinks it can do with our bodies, bearing in mind this is Gates' computer company. Moderna claims they can program people like computers. So to conclude and wrap this up, back to Harold Hillman, who said, "And I think we have pretty much covered all that here, yes. Now if they can't make any of this stuff work, do you really think they can make transhumans using this technology? I would take a stab at no. Which is why I'm not delving into the nanotech at all. It's all drawing board stuff, but this experiment only concludes they can't make it work in practice, so it's a mass cold dressed as a vaccine for a manufactured fake disease. If they can sterilize some of us, that would be an added bonus, but I doubt it is caused by their genetic experiments. It's just a side effect of the poisoning."